right, good morning, everyone. So can I ask you, good morning, any questions from last time? And again, I'm just struck by the stench in this room, so I apologize, really. I think all of us need to make complaints about this, if you will. Back me up. I'm going to call facilities management, but you can make complaints to the department head, Francisco, and if all of you do it, we'll get some action and change this room. It needs to be addressed. I have no idea. You poor people that are stuck in this room, I don't know how many classes you have here, but it's getting worse, it seems. I'm not sure. There's not enough airflow in here, and it's a problem. So that said, I realize you're dealing with a little bit of barrier to paying attention to me. So, no questions. Then I'm going to move on. We have a lot to cover today. And we're going to begin and end with talking more about glutamine. So I'm going to introduce the fact that glutamine metabolism takes place in two different locations in the liver. We're going to talk about other tissues where glutamine metabolism happens. But, you know, I told you glutamine is one of those really critical amino acids because it carries amino nitrogen around between tissues, right? So it's really one of the most important amino acids in the body, and it's very much so in circulation. But in the liver, we've talked about, and I need to make sure I'm looking at the right, so we've talked about the periportal hepatocytes where glutaminase enzyme is acting to release ammonia to feed into the urea cycle, and that those two enzymes are coordinately regulated, right? So when the urea cycle is cranking, so is glutaminase. And so glutamine is coming in from a lot of different tissues. We're going to talk about muscle as one of the more important ones. Whoops, this is moving. Wow. Pardon me. But the other thing you need to know, so this is the periportal hepatocytes, downstream as the blood is leaving the liver, so this is written as if it's right next door to each other, but of course it's not. Another set of cells, the paravenous hepatocytes, can make glutamine. Okay, so I'll just tell you that's what's on your slide, of course. But what we need to back up and just say is that the liver has two primary ways of dealing with ammonia, which is toxic to the body, right? So it's important that we have multiple ways of dealing with it. The first way we've talked about is urea synthesis, right? Urea is detoxifying ammonia. And that is, again, primarily any ammonia coming in from the intestine is going to be made into urea. Glutamine coming in is going to be cleaved and yield ammonia and make urea, right? So that's the primary way. The second way is that any ammonia that might escape, and there are regulating things that will make ammonia pass through and move along and get taken up, or let me finish this thought, sorry. Any ammonia that escapes being made into urea is going to be taken up very avidly by the perivenous hepatocytes. Okay, so that's kind of a backup plan or a catch-all. Any ammonia that doesn't get made into urea will get made into glutamine by the perivenous hepatocytes. And there are differing levels of activity of these two enzymes in these two locations depending on different factors that we're going to talk about. We'll end talking about much more detail about the physiologic conditions under which the liver is primarily breaking glutamine down with its glutaminase enzyme over here in the periportal hepatocytes, and which conditions are these cells actively taking up ammonia and making glutamine, and they're different, okay? So we're going to talk about how important the liver is in inter-organ glutamine metabolism. So let me just reiterate here. So you've got the two enzymes, glutaminase, and we know about that reaction, and glutamine synthetase, right? And they're happening in two different cells, very different locations. And if you think about this, one of them requires energy. To make glutamine, it takes energy, right? So I asked this question, how does the location of these two enzymes in two different cells help us to avoid a potentially futile cycle? Have you heard about that term before, that phrase? Potentially futile, futile cycling? Anyone? Okay. Well, what I mean by that is energy wasting. Futile cycling is energy wasting. And the body doesn't like to have a lot of futile cycling going on, right? It doesn't happen. We have a lot of stop gaps for that. So if you think about it, the fact that these are in two completely different locations, and actually I'm going to go back. It's always hard to go back. I have to hit that a lot. Interestingly, and these are really, they're drawn not to scale, obviously. Glutaminase is a mitochondrial enzyme. Glutamine synthetase is a cytosolic enzyme. But they predominate in totally different cells as well. So there's a subcellular, the fact that one is a mitochondrial enzyme, one is a cytosolic enzyme, is one level of compartmentalization, although note that they're really not in the same cells. And then the more macro and important, physiologically relevant one is that they're divided by cellular compartment within the liver, right? So they're in totally different cells. So you can see by that separation, it avoids just making glutamine using energy and then breaking it down. If they were in the same location, that would be a futile cycle going. Okay, is that clear, everybody? Way up there in the nosebleed section? Okay. All right, now I gotta, sorry, and here's a liver lobule. This is the basic structure of the kidney, of the liver, sorry, where the portal area is distant by each of those, these are hepatocytes, right? And then we've got the paravenous hepatocytes over here. So they are distant in location, and here's a kind of again, also abbreviated, but you can just see that there's a distance between them. They're separated by compartment, and that is very helpful. All right. So here's a little more, just showing you the structures involved, and it's glutaminase and paraportal hepatocytes. And then in paravenous hepatocytes, you got glutamine synthetase. Now, add this to your information about glutamine synthetase. And this is also hopefully makes good sense to you. But this enzyme has a low Km, which means very high affinity for ammonia. Low Km means high affinity for ammonia. So again, any ammonia that gets presented to those paravenous hepatocytes is going to be avidly taken up and converted to glutamine. That's what that means. So it's going to, any ammonia that it sees, those paravenous hepatocytes are going to make into glutamine. Okay, so that is enough for this moment. We're going to get back to glutamine again at the end, and actually I'm going to tell you a little bit more as we get through each tissue after we talk a little bit more about the fate of an amino acid carbon, because I need to just kind of wrap that up again, revisit that. So you know that anything that we talk about, and we usually talk about it in as extreme cases as possible because the nuance of overlap between fed and fasted is makes things a little more difficult to understand, right? So I'm presenting to you the fate of an amino acid carbon after the amino group is removed. And actually this is holistic overview, but we're going to talk about the fed and then the fasted state and a little bit of overlap, and I'll explain as I move through it. So first question, in the fed state we ask, is carbohydrate energy adequate to meet the most basic 150 gram carbohydrate, is pretty much needed to meet the brain's obligate need for glucose. So the question is carbohydrate energy adequate? And if the answer to that is yes, in this state, of course, we're talking about the hormonal milieu of high insulin, low glucagon, typically. Then we ask the question, are amino acids in excess of needs for protein synthesis? If no, but they're adequate, you've just got enough protein there, we're going to use what we need to for protein. And then any excess, this is a difficult diagram to follow, I realize, but any excess after we use what we need to for protein synthesis, we're going to take the amino group off and we're going to use that carbon skeleton, right? So that's what this is trying to show over here. We got urea, primarily the form of nitrogen that we're excreting is urea in the fed normal physiologic state. And then that alpha keto acid will be used for these purposes, energy, fatty acid synthesis, potentially glycogen synthesis, okay? So that's the fed state. Any questions about that? All right. Now we can come back over here because I've animated it this way, I think. No, I'm not going to come over there. Not yet. Hold on. I know it's on your slide, but let me try to keep this sensible. So now we're going to talk about the fasted state. In that state, you're going to have low insulin, high glucagon, right? Low insulin to glucagon ratio. Yeah, it's animated. Okay. I'm going to just click through this so I can, yeah. So let's say in the fasted state, amino acid will be degraded, deaminated, I have drawn this, and I'm going to explain this more as we get ending up in glutamine metabolism. I'll talk more about how glutamine synthesis becomes a more important way of getting rid of ammonia than urea, relatively speaking, in an extended fast, okay? So it's a little confusing to say that here, but it will make more sense as we move through the various tissues. But just know that urea synthesis becomes relatively less important in a fasted state for excreting nitrogen. We actually make glutamine as an important way of getting rid of nitrogen. And the keto acid of the amino acids will then be used for glucose. They'll be converted to ketone bodies as needed, and they'll be used for energy in particular tissues, right? So that's the fasted state. Now what this is showing over here is carbohydrate energy intake adequate. If the answer is no, that would be somebody who's really restricting carbohydrate, but they're consuming protein, right? That'd be somebody following an Atkins or a South Beach diet or whatever, where you've got, below that threshold, the ideal of 100 grams or whatever, and maybe even just 10 or 20 grams, and then this pathway or this side of the box would be more relevant in that condition. Where are the amino acids coming from in a fast? Predominantly. Okay, endogenous sources like skeletal muscle, big source of amino acids, particularly early in a fast, okay? So when I have fasted, I'll try to call out the nuances of this, but I've left this as that right after you've depleted your glycogen, so it's around 24 hours to 48, first few days of a fast, you're going to be really using a lot of skeletal muscle. Over time you adapt, and we you know, we talk about that. I'm sure that Dr. Chen talks about that as well. But so we're going to kind of consider this kind of early fast. You're going to have skeletal muscle supplying a lot of those amino acids. And of course, in this case, you've got exogenous, right? Your dietary coming in. Okay, let me just make sure I haven't missed anything that I wanted to call out. Remember this, and you know, just remember that you can't make glucose from acetyl-CoA, right? That notion of anaplerosis means that you are adding carbon to the TCA cycle to allow for glucose to be synthesized, right? And so leucine, remember we said, only yields 2 carbon intermediates, acetyl-CoA and acetoacetate. I'll show it to you in a moment when we get to muscle, okay? So leucine cannot be used to make glucose. It is unique. That's what we mean when we say it's uniquely ketogenic. And just, again, you can't graduate without just knowing that you can't make glucose from acetyl-CoA. That's just a, you know, 2 carbon unit entity. You cannot make glucose from it. So that's what we mean about leucine. Are there any questions about that? And we talked through that before, so I just wanted to make sure that that's clear. Okay. So we're going to stay in the fed state as we talk through this slide. And so I had, at the beginning of the liver section, I'd said that about 60% of the amino acids coming in the fed state through the portal vein from the intestine are going to be catabolized, at least to some degree, right? They're going to be transaminated, most of them. They'll be deaminated, you know, the amino group will be taken off. So what happens to the rest of them? I also had said that about 20% are going to be left to go unscathed onto other tissues. And the ones that are predominantly in that pool, about 70% or so of those are going to be branched chain amino acids. Okay, so branched chain amino acids make up the bulk of what leave the liver in the fed state. So guess what? In the circulation, concentration of branched chain amino acids goes up quite markedly after we eat. The other amino acid concentrations fluctuate a little, but not nearly to the extent that branched chain amino acids go up. And it's, you know, stays up for a few hours after we eat. Now, I'm going to say this here, and then we're going to get back to it when we talk about the muscle and metabolism of branched chain amino acids. But a major reason, I mean, you might ask that, right? Isn't that a little curious to you? You know, why is it that the liver just allows so many of those branched chain amino acids to leave the liver? So anybody want to take a guess as to what the answer might be? Yeah. Yeah, okay, so great, you're thinking very well, right? There's some enzyme reason why maybe other tissues have enzymes that would help break it down and the liver might not, right? So if it's an amino acid, what do you suppose, given the rule that I said about amino acids, what kind of an enzyme is probably lacking or at least has a low activity in the liver? Knowing what we know about amino acids, right? All but what? Lysine and threonine undergo transamination. So it's got to be an aminotransferase, right? That is specific for branch chain. It's called BCAAT. I'll write it out on another slide, so I'll just leave it at that for the moment. It's a branch chain amino acid aminotransferase. That activity is low in the liver, and that unilaterally explains why the liver allows those branch chain amino acids to move on to other tissues. Okay, so that's an important regulatory thing. Low BCAAT activity. All right, and then about another 20% are used for protein synthesis in the liver, and that's highly important, and Dr. Chen will be talking about protein synthesis, I'm pretty sure. Okay, So now, we're ready to move on to the next tissue. We've got a few to go through today. So I need to make sure I'm, oh, and I remind you, this is, review for the test. Which amino acid is the most highly conserved because it doesn't undergo transamination as the first, second, or even third step in its degradation? Do you remember? It's one of the essential. Anybody? Yes, thank you. Lysine. Great. I don't think there's anything surprising in that summary, so I'm going to, in the interest of time, I'm going to let you use that. Oh dear. Sorry. Jillian had a field day with my she's like, Mom, it's just too boring. How do you keep them awake? Okay. So now we're going to talk about the intestine, amino acid metabolism in the intestine. And I've given you some objectives that help us kind of focus a little bit, but I'm going to get us through. So now we're going back, right? So we talked about the liver. Now we're backtracking a little after a meal, right? Because the intestine is where the amino acids are coming in. You've undergone digestion and absorption. So we're going to stop here. And just think about the fact that, of course, the intestine is going to need to retain some of those amino acids that come in. It's not entirely philanthropic and, letting everything go on through the portal vein. It needs to do a fair amount of important things with amino acids, right? So can you think about what lipoprotein would probably be the most important in the intestine and what apoprotein would be involved there? What lipoprotein is important to be synthesized for the intestine? Helps us carry and absorb, helps us absorb fat, right? Chylomicrons, right? Chylomicrons, go into the lymphatics, that's fat metabolism, but anyway, it's an important thing that the intestine can synthesize chylomicrons. So it's got an apoprotein. Does anybody remember that one? Remember? B48. So ApoB48 is the lipoprotein, the apoprotein that is needed for chylomicrons, and that's synthesized in the intestine. We have a lot of GI hormones, and you probably, if you read anything lately, you are probably aware of ghrelin and PYY and all these peptides, hormones, right, that are produced in the gut. And then we've got pyrimidines, purines, for DNA and RNA and other things like that. So there are a lot of nitrogen requirements within the intestine. And then this very important thing right here is energy, actually. So the intestine, we talked about this, I know, I always feel like I'm a broken record about this, but there are, which amino acid is preferentially used by the gut for energy that we talked about? Glutamine, right? It's going to be the answer for the day. We're talking a lot about glutamine. Not the only thing, thankfully, but I will talk a lot about glutamine. So primary energy source, so let's talk about the various sources. So when you're in the fed state, dietary glutamine, well, just to make it simple, I'm going to say that it's primarily glutamine, but it will also, the intestinal cells can also use its cousin glutamate and aspartate. So GLU and ASP to a far lesser extent. So it's, for our purposes, you can just think of it as glutamine, if you'd like. About 40% of the energy needs for the gut come from glutamine. Aspartate and glutamate as well. Okay, so about 40% coming from diet. Blood glutamine, right? Remember I told you it's the most important, it's the most abundant amino acid in circulation. So blood glutamine provides about 40% of energy, and then about 20% of its energy comes from glucose, okay? So glutamine is, about 80% of the energy requirement. Now, That, of course, is going to change the ratio of that diet versus blood. If you're in a fast, you'll get more from blood, okay? But just in the normal, when I say normal physiologic state, I mean the state where you're eating at regular intervals, right? You'll get about 40% from diet, 40% from blood, and then 20% of the energy comes from glucose. Is that what you were going to ask me to repeat? Okay, so that accounts for 100% of the energy requirements. So big, big part of it coming from glutamine. Now, where does the blood glutamine come from? We're going to talk about this. Skeletal muscle is a huge supplier of glutamine for the gut. Where else might it be coming from? And I haven't told you anything about any other tissues. We've talked about muscle, but where do you think it might also come from? We talked about 4 or 5 minutes ago. Where? Sorry, thanks. Not urea. Where? The liver, right? Paravenous hepatocytes. So they've got glutamine, the machinery for making glutamine. The lungs also can do it. But the vast majority comes from skeletal muscle under kind of what we would say the normal physiologic state where you're eating and at regular intervals. So again, we're gonna get more nuanced in our discussion as we move on, and at the end we'll talk about very different conditions where that might change just a little. Glutamine has a huge trophic effect. What does that mean? What? Say it louder, will you? Okay, trophic, growth, right? So the mucosal lining of the intestinal tract turns over every 24 hours, right? So giving glutamine actually nourishes the gut, helps it to do that, provides its energy that it needs, and has a trophic effect, okay? And then what enzymes does the intestine need to metabolize glutamine? I'm going to show you on the next slide, so we'll answer that, and if it's not clear after we talk through it, then you can ask me again. Okay, this is a bit of a busy slide. I want to just give you the key down here. Purple is the compounds that are produced from glutamine, and the sunbursts are the things that get released from the intestine. So where do we want to start? Well, let's answer the first question. We said which enzymes are needed. So glutamine metabolism, remember, huge amounts of glutamine taken up by the gut and used, right? So the gut needs to use it for energy, right? So in order to do that, this hopefully, I will say this now, because if I forget to and we're finishing up. For the exam, you must not just try to memorize blindly, okay? You want to use your best brain about thinking through the rationale. When we ask about enzymes, don't just blindly memorize them. Just keep in mind what the tissue is doing, and hopefully it will help you be clear about what enzymes are needed, right? So if you need glutamine, you're going to use glutamine for energy, then that means you got to get it down to its component parts, right? So the first enzyme has to be glutaminase, right? So glutaminase will act on it, and lo and behold, we got one of our products and one of our export products. I mean, it's essentially an export product, is ammonia, okay? So that ammonia, where's that? What is that going to be? What's going to happen to that ammonia? Goes in through the portal vein to the liver. What's mostly going to happen in the fed state, particularly? Urea, right? Eureka, urea. Okay, sorry, that was really bad. Haven't had nearly enough coffee this morning. Okay, so then we've got glutamate, right? Glutamate is left there. Now what? What do we got to do with glutamate? If you got to get that down to its carbon skeleton, actually you're not going to get it to its carbon skeleton exactly through where I was thinking my head was going, but ALT is the enzyme that is predominantly available, and it makes alpha-ketoglutarate that will enter into the citric acid cycle and produce ATP that way. So that's really pretty much how we get energy from glutamine, is this transamination reaction that will yield alpha-ketoglutarate. And in the action of ALT, it's producing alanine, okay? And then that's also one of the major export products. So alanine, the liver likes alanine, will take it, do whatever it needs to do in terms of, gluconeogenesis to some degree, even in the fed state, but it's certainly important in the fasted state as well. Okay. What else to say about glutamate? Glutamate, I'm showing here, these are kind of sidebar, just so that you are aware of these things, but I'm not probably going to ask you about proline, which is another non-essential amino acid. Remember how there's a lot of interchangeability between non-essential amino acids, so that's what that's showing. Glutathione is just one of those things. Does anyone have any clue what glutathione is? You have ever heard of that? Sometimes if you go to GNCs, or people that go to GNCs, they'll take various things. But what do you think about it, Jonathan? Any? Yeah, it's the component that is essential for synthesizing one of our important antioxidant enzymes, glutathione peroxidase. Actually, a little bit of trivia, it's, basically it breaks down hydrogen peroxide in the body. And our immune cells release hydrogen peroxide when we're trying to blast viruses or, blast bacteria, right? And essentially it's toxic, so you got to have a way of dealing with it, and glutathione peroxidase is one of those ways. And so glutathione is a tripeptide, and glutamate is one of the three that are used to make it, okay? And that's all I'll say about it. So focus here. So glutamate is getting converted to ornithine, which then gets converted to citrulline. So I want you to think about what enzymes and what sequence of events do you need to do that? Where have we seen ornithine before? The urea cycle. So guess what? There are some of the enzymes from the urea cycle in the intestine. Okay. Now let me just think about, I'm going to show you it in a moment here. Let me just think about where I want to. Yeah. Question? Did I hear up here?
Yeah, so we previously wrote down ornithine and citrulline in the urea cycle.
Yeah, hold this thought. Hold the thought, okay? Just a moment, and I'm going to explain, all right? So the fact that this is happening is really important. You need to know how it's going to happen, and I'm going to show you on the next couple of slides here. I think I'm 2 slides away at this moment. But know that citrulline is a really important product as well. So we've got alanine, ammonia, and citrulline being exported from intestinal metabolism of glutamine, essentially, okay? All right, so here's our summary. What happens to the ammonia as one of the products? And my slides are always, they're in the order they're in. I want you to begin thinking about how we need to convert ornithine to citrulline. You think back for a moment, go back if you want to, I don't know how far back in your notes. Take a look at the first part of the urea cycle slide. It's maybe what, 6 or 8 slides back. And maybe some of you have already internalized and your wheels are starting to spin. In order to get from ornithine to citrulline, what is the, so looking at that slide, right? You've got the busy slide probably of the urea cycle up in front of you. What enzymes do we need in the intestine in order to get from ornithine to citrulline? Okay, I'm going to need hands so that I can, okay, let me just, so that I was ask you. Yeah. Okay, so we need ornithine transcarbamylase and what other substrate to combine with ornithine? Yeah. What is it? Carbamyl phosphate, right? Carbamyl phosphate. Oh, okay, how are we going to get carbamyl phosphate? We need that as well. So what other enzyme do we need? And substrates. So, Jonathan? So we need CPS1, carbamyl phosphate synthetase, and lo and behold, ammonia is one of the substrates. Lucky for us, we got a lot of glutamine around that we're releasing ammonia, right? So the ammonia, some of that ammonia is going to stay in the intestine to be used for making carbamyl phosphate, because we need it in order to get to citrulline, right? And then I also said, what else happens to the ammonia, right? We said it goes to the liver for urea synthesis. And then under certain conditions, some of it's going to be taken up by the paravenous hepatocytes in the liver to be made into glutamine, potentially. And again, we're going to talk about that later at the end. Okay, so certainly some of it's going to be used to make carbamyl phosphate. Some of it will be used for urea, potentially some of it will be used for glutamine in the liver as well. All right, how about the alanine? That's another product. And I'll just say this again. It goes to the liver where it might get transaminated, might get used for making glucose, depending on what the physiologic state is. Might get oxidized as well, right, because pyruvate. And then we've got ornithine. Now, remember, this is a product, not an export. Ornithine doesn't go out. We've answered this, right? We've seen it before in the urea cycle. What compound is made from ornithine? Citrulline, right? And the enzymes that are required, you've got to go back and think about that. You're going to know the urea cycle really well. It's not the whole urea cycle. I'm going to talk about that just next on the next slide. But you think about, we said in order to make that citrulline, you need the CPS1 enzyme, you need the ornithine transcarbamylase enzyme. And what do we know about CPS1 activity? Say it. We need NAG, don't we? Dang, right? So we need another enzyme to get that CPS1 active. What enzyme makes NAG? Remember NAG, that allosteric activator of CPS1, without which magnesium can't bind to the ATPase and it can't work? Acetyl glutamate synthetase. Now, while we're on this, I know this is a lot on this one slide, but if you think about acetyl glutamate synthetase activity, you might remember what activates that enzyme from the urea cycle. What condition helps us to increase the activity of acetyl glutamate synthetase? You say it louder? Well, there's a certain concentration of a certain amino acid that will drive acetyl glutamate synthetase activity. Arginine, right? So when is the intestine really focused on making citrulline? It's the fed state. Okay? So that's a really important thing too. So all of this hopefully comes together and makes sense to you, that, you know, you have to have a high arginine concentration to get the acetyl glutamate synthetase action, to get the CPS1 action, to get the citrulline made. Okay, so it's in the fed state when that's happening. Where does it go when it leaves the intestine, and what's it used for? So again, this is the product we're talking about, citrulline, and what's it used for? So let me explain that. So underline partial up here, okay? You need to know that not all of the urea cycle enzymes are in the intestine, and that's important because the intestine is not making urea, right? Only the liver makes urea. But it's really critical that these enzymes are here to make citrulline. And what happens to it is the citrulline will bypass the liver and it'll get taken up by the kidneys. And this is an example of interorgan cooperativity. Now, what this allows for, and I'm going to show you on the next slide, okay? So this is the first part of the story. The intestine making citrulline. Citrulline passes through the liver, doesn't do, isn't entering into the urea cycle there. Goes to the kidneys, only in the fed state, okay? This is the fed state. And the reason for this is that in the kidneys, that citrulline will be used to make arginine, which can then be released from the kidneys to be used by other tissues when we want to be making protein. What happens to the arginine in the urea cycle? Okay, so this is now, I'm moving to the kidneys, right? So I just said citrulline bypasses the liver, it goes taken up by the kidneys, and that allows for whole body protein synthesis from the arginine that gets released from the kidneys. If we were in the urea cycle here, this is written wrong, this is written for the kidney here, but just remember back for a moment. The last step of the urea cycle in the liver is that arginine gets cleaved to yield urea and the cycle continues, right? If you had all the machinery in the urea cycle, the whole purpose is that last step we cleave arginine to yield urea. So guess what? Not a lot of arginine leaves the liver in the fed state, because it's really in the urea synthetic, as that's cranking, because you've had a lot of protein in your diet, right? You're going to be cleaving that arginine to yield urea in the liver. So we need another way for tissues to have access to arginine, right? Because the liver is a big hog, it's a big first pass metabolism. It's going to do what it does. It's making urea, and that arginine doesn't make it out to the other tissues to any significant extent. So we need this interorgan cooperativity between the intestine and kidneys to allow for arginine to be synthesized elsewhere other than the liver. Okay? So now I'm just showing you this right now. We're going to talk most about it here. I'll revisit it in kidney just so you can remind yourself that it goes with the kidney material, okay? But it's important that we talk about it now. So the kidneys are taking in citrulline coming from the intestines, and it's got the machinery to combine citrulline with aspartate to make arginosuccinate. It's got the arginosuccinase to release arginine. And it has a very small amount of arginase activity. And this is where students usually go, well, that doesn't make any sense. If you've got arginase, how come we don't just keep making urea at the kidney? Well, guess what? Ornithine's got nowhere to go in the kidney, really. It doesn't have the machinery to recycle it, to get it over back into citrulline, it's not present in the kidney. So if you got a little bit of arginase acting on arginine, you might get a little bit of ornithine made, but that's going to just shut down that enzyme. So arginase, for all intents and purposes, is not active in the kidney. If it were totally paper biochemistry, I would say it's not present, but we know it is, right? So it's just very small amounts. Nothing ever works out quite perfectly in real life, right? So, but it does work out perfectly because that enzyme, it doesn't have any activity. So you're not making urea at the kidney. Please put that in big, bold letters on this slide. We do not make urea at the kidney. What we do make is a lot of arginine from citrulline, because we have these two enzymes. We can recycle fumarate because those are abundant in every tissue, right? So we need all those. I think I've got it animated here. So we have the enzymes that allow us to recycle fumarate, which is useful. but we don't have essentially arginase activity. So arginine then gets shipped out for whole body protein synthesis. What questions do you have about this? Yes?
So you said that arginine drives the acetylglutamate place, but then in the end you're making arginine. Why doesn't that active arginine drive this process?
So we're in different tissues. Now we're in the kidney. Okay, please note, I don't have a picture of it, but we're now in, this part of it is happening in the kidney. Okay, so the kidney only has these enzymes. The intestine only has these enzymes. Okay, so in the intestine, fed state, arginine concentration is good, right? It's in protein. That'll drive acetylglutamate to make NAG, so we get CPS1 activity, et cetera. Okay, but now I'm moving to the kidney. So citrulline, we're going to the kidney here. Don't think about arginine concentration and CPS1 because it's irrelevant here. CPS1 is not present in the kidney. So we're talking localized regulation of enzymes. It goes to other tissues where it's taken up, okay? Oh, some of it probably, yeah. So you know, arginine concentrations, yeah, that's one maybe a bit of I've not seen exactly what that tends to be. All the other amino acids tend to kind of be maintained in a sort of relatively stable concentration, fed versus fasted. I mean, that's one thing. So sure, if some arginine, you know, but I don't want you, don't think that way, okay? I mean, if you try to make it, to understand this, it's easiest if you just think about it in terms of the cells, where these enzymes are, and what is influencing their activity. That's just, of course, there's going to be some gray areas, okay? So arginine, you know, in the gut, you know, that you're taking in, you know, possibly some of that is going to escape and go through circulation to the kidneys or to other tissues, right? But for the most part, there's going to be a fair amount of it extracted by the liver and used, you know, in the urea cycle, you know, basically produced in the urea cycle and then cleaved to urea. Okay, any other questions about the fact that neither of these tissues, so intestine and kidney, don't have the urea cycle, the full urea cycle. They've got partial enzymes. Okay, neither of them produces urea, just so you just keep very clear the liver is where urea is synthesized. Are there any other questions as we're talking about this before we finish with the intestine? Then I'll let you break and then we'll go back, we'll start with the muscle. Any other questions? Okay, take a break, go get some oxygen. Let's come back before, can we, because I'm like 2 minutes, can we do 10 minutes? So come back before 20 till, is that too confusing? You know, like 2 minutes before 20 till or so. Just take 10 minutes. Go do what you need to do and come back. All right. Hello. Whoops, I'm not on. Thank you for coming back so efficiently. We're going to press on because we got some tissues to cover. We got muscle, we got kidney, and then we got inner organ glutamine metabolism, all to do in our last hour. Not even hour, right? Now, so we've just been in the intestine. We were briefly in the kidney, okay, but we'll just because I had to explain that. But now we're moving on to the skeletal muscle, okay, massive organ in the body if you think of it in its, oh gosh, and I'm doing it again. Hold on. So we're going to talk about branched chain amino acid metabolism predominantly and what comes from that in the muscle, where it goes, et cetera. So here's our very abbreviated roadmap, right? We were in the intestine. We've talked all about the liver. Now we're in the skeletal muscle as one of the peripheral tissues, right, where amino acid metabolism happens. We've already talked about the answer to this question, right? Which amino acids are the primary ones that are bypassing the liver to come to the muscle? Ones that the liver lets go unscathed, 70% of those being what? Branch chain, right? Now, other amino acids in constitutive concentrations are also making their way to the muscle. And the amino acids that arrive at the skeletal muscle, including the branch chains, are going to be used for, what do you think? So we talk about in the fed state, what's a big primary important thing about metabolism in the fed state? For protein anyway, what would you be doing? What hormones are high in the fed state? I mean, what's the hormonal milieu? High insulin, low glucagon, right? What does insulin drive? Synthesis? Did I hear? Sorry, everybody talking at once. It's like, shoo, 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 you know. Protein synthesis, right? So those amino acids are going to be used for protein synthesis. Some of them might be catabolized depending. But majority we're going to be, doing maximizing protein synthesis. These are the structures of the branched chain amino acids, hence the name. You can get the idea. Hopefully you'll remember my little antic with the valine V, you'll remember it's branched chain. And then we've got two primary enzymes that are involved in the metabolism of branched chain amino acids. The first step is a reversible transamination. All transamination reactions are reversible. And it's catalyzed by the branched chain amino acid transaminase or aminotransferase, depending on the source that you look at. So this is a very global, I'm not giving you each of the keto acids right here. I give it to you later in the slide set. So just remember, PLP, what's the active, what does that stand for? Pyridoxal phosphate, I think I heard. Not really. I heard mumbling. This room is horrible for that. What is the vitamin? Vitamin B6. So PLP is the active form of vitamin B6, so it's going to be required for those. Incidentally, BCAAT is abundant. It's in the mitochondria and the cytosol, just like any other aminotransferase. So both subcellular compartments. And then the second enzyme is the BCKAD, branched chain keto acid dehydrogenase. It's written out on the next 2 slides from now, so don't worry about that for now, BCKAD. This is irreversible. I've got the arrows because there are a number of steps after the BCKAD, okay? So what it does is it'll act on the alpha keto acid of those branch chains. This is a mitochondrial enzyme only. And these are the end products of each of the branch chain amino acids. And remember what we meant when we said ketogenic, uniquely ketogenic. Actually, I really should keep that word there, uniquely, because any of them can be made into fat, right? But what is true about leucine is that you can't, it's absolutely not glucogenic. It's only ketogenic. Can't be used to make glucose. Yeah.
Why are there 3 different products if they're all being converted to alpha-keto acid?
Well, they're different things,
Alpha-keto acid
right?
is generic.
Yes. Yes, sorry. I think I have it later. I'll give you what the alpha keto acid is, perhaps. I can't remember if I do that, but yeah, know that this is generic, right? I'm just giving you the generic here. Okay, let's talk a little bit more specifically here. So BCAAT, branch chain amino acid transferase, cellular location, both the cytosol and the mitochondria. Like other transamination reactions or other transaminases. The type of reaction, reversible transamination. Just to call that out. It's transamination, but know that it's reversible. What about this? BCAAT activity in the muscle compared to the liver. What do you think? A lot, say it louder, will you? It's not active in the liver, so in muscle it's a lot higher. So you can say much higher in the muscle and pretty much non-existent or not active in the liver. Now, before I get to the next thing, the significance of that, the fact that you don't have activity in the liver, the significance of that, it allows the branch chains to leave the liver without being transaminated, right? So they come intact to the muscle. That's the significance of the fact that liver has so low activity, essentially none. It allows for those branch chain amino acids to leave and be taken up by the muscle. So, what happens to the alpha keto acids of the branch chain amino acids I've got on graphic slides. Okay, so you're going to need to know the answer to that, but I'm going to give it to you in a more organized fashion than just calling it out here. Okay, second step in branch chain amino acid metabolism is this BCKAD. There it is written out. Cellular location, mitochondria. What type of reaction is it? Irreversible, and I didn't say this before, it's a decarboxylation reaction. Oxidative, in fact. Oxidative decarboxylation. If you want to stick oxidative before that, it's the same kind of enzyme as pyruvate dehydrogenase, which I know you've heard about, and I'm not sure how much nuance you've learned in terms of the cofactors. Did you kind of learn a lot about PDH? Did you know that it's an oxidative decarboxylation reaction? I'm not seeing any nods, so I'm not sure. Well, anyway, it is. It is an oxidative decarboxylation reaction. You know, it's highly similar. It has the same kind of 3 subunits. And again, depending on how much you've learned about that, I won't kill you with the details on that. But I'm wondering if you remember how PDH was activated. Was there a phosphorylation reaction involved? Do you remember at all? Okay, again, I'm not seeing any shakes, nods. Okay, I will tell you, let's just focus on BCKAD and you'll know that they're very similar to PDH, okay? In order for it to be active, it needs to be phosphorylated. And that's true of a lot of enzymes, actually. Phosphorylation is sort of a way of activating them. So in order for BCKAD to be active, it needs to be phosphorylated. There is a kinase. Have you heard that term before? Kinase. So kinase, there's a BCKAD enzyme kinase, and I'm not sure exactly what it's called. It's probably BCKAD kinase, but don't worry about the name. And that helps this enzyme be active. And it's active, much, much more active under conditions when there's low insulin. Okay, so the BCKAD enzyme is active when essentially we've got low insulin around. So I'm going to go ahead and click here. The necessary cofactors are on about 3 slides from here, and they are the same cofactors that are required for pyruvate dehydrogenase. It's a bunch of vitamins. So you can imagine what a field day I had when I was teaching back at Virginia Tech, because I got to teach vitamin and mineral and protein metabolism, and I got to do a lot of big picture stuff so that you would figure out how thiamine is involved, or niacin, or riboflavin, right? So I don't think I really go to town and ask you that on the study guide, so if I don't worry about it, but it's nice to know that there are B vitamins involved in amino acid metabolism. It's an important thing for you to appreciate, I think. BCKAD activity in the liver compared to the muscle, and under normal circumstances, normal physiologic conditions, when you're eating and every 5 or so hours, you can say that BCKAD activity under normal circumstances is way higher in the liver than it is in muscle. So that would be like, if we're talking fed state, okay, it'll be much higher in the liver than muscle. So hopefully that meshes with what I just told you about insulin, right? BCKAD activity increases a lot when insulin is low. Now, so that explains starvation, right? Starvation, low insulin, you're going to have you'll activate the kinase, you'll activate that enzyme. What about metabolic stress? Now, those of you that are not with me in MNT probably have no idea what I'm talking about. You're probably thinking, yeah, we got an exam coming up. That's metabolic stress, right? It's actually not that, right? Does anybody from MNT want to explain kind of what we think of as metabolic stress? Under what conditions might you be in a metabolic stress response? You got to raise your hand and speak out. Marie, can you do it? So the metabolic stress response, as we talk about it, happens when we are undergoing massive injury, not just cutting your finger, but falling off a roof, being hit by a bus, God forbid, something like that. Or when you undergo major abdominal surgery, open heart surgery, your body mounts a metabolic stress response, okay? Under these conditions, you could also have a massive blood infection, sepsis, something like that. Metabolic stress, when that happens, we have what happens, we call it insulin resistance at the peripheral tissues, and muscle is a big one. Insulin resistance means that your skeletal muscle doesn't really recognize the insulin. It's not really acting at the tissue very well. So in starvation, where you actually have low insulin to glucagon ratio because you're just not producing, there's no reason to release insulin, and metabolic stress, I really am loose in my language here. You might write metabolic stress, and if you want to say to injury, that would maybe help you really picture what we're talking about. We're talking about ICU patients, intensive care, right? Critical illness. In that condition, you also have low acting insulin at the muscle because we have insulin resistance. So under both of these conditions, branch chain ketoacid dehydrogenase activity increases 5-fold. Way big increase, arrow up 5 times, whatever you want to say. Branch chain ketoacid dehydrogenase activity increases something like 5 times normal in starvation and metabolic stress to injury and major surgery and critical illness. And that's important, and I'm going to show you on the next few slides why that's important. Yeah? No, I don't want you to think about metabolic stress as a whole body systemic condition, okay? I'm not hearing you, Lena.
The branch chain alpha acid dehydrogenase is still primarily in the liver, even
No, it is in the liver, but BCKAD activity under metabolic stress and starvation increases 5-fold in the muscle. Sorry, didn't quite get the main of your question, okay? So in muscle, it's really increased that activity, and I'm going to show you in a second how that works and why it's so important. First, let's just, I've got this here because it's something else that you should be aware of, that you can have defects, inborn errors of metabolism, particularly for amino acids. There are a few that we've talked about, PKU. This one is related to a defect in the BCKAD enzyme. So if you don't have adequate, the gene that basically doesn't allow you to make BCKAD, you end up with a disease called maple syrup urine disease. And guess why they call it maple syrup urine disease? The urine smells like maple syrup. You know, for Minnesotans, this is kind of our disease. No, I'm sorry, that was a bad. I'm reaching, it's hard. Keto acids build up in the urine, and so do the branched chain keto acids. Also, branched chain amino acids will build up in the plasma and the urine. It leads to neurologic problems, mental retardation if we don't deal with it. Within 10 days of birth, someone will die, a baby would die. So we screen for it. If we know it's present, we give them specialized formulas, just like we did with PKU, where we're giving low branched chain amino acid formulas, and we just provide what's needed to do protein synthesis, but no other than that. Okay. Now, I've got two figures. At first glance, they look a little busy. On your slides, they look a little busy. So we're going to talk about the fed versus the fasted state in muscle. So let's start up here. Fed state, branched chain amino acids coming in to the skeletal muscle through, active transporters, come in. They undergo the branched chain amino acid transaminase reaction and yield the BCKADs, branched chain amino acid alpha keto acids, those ones we talked about. Those are going to be released in the fed state. In that transamination reaction, you're producing glutamate, which then can go through the ALT reaction to make alanine. So alanine and the BCKADs are going to be export products from the muscle. And then, lo and behold, we talked about the muscle being a primary producer of glutamine for the intestine. And so it's got glutamine synthetase, and I'm not showing the entire reaction, so I'm missing ATP and all the other things, okay? So just this is an abbreviated reaction to make glutamine. And glutamine and alanine are going to be released. They'll be taken up in large part by the liver, but also they'll make it to other tissues as well. And then over here, we've got what's happening to those alpha keto acids. Other tissues like heart and the kidneys may take those up. They have transaminase. The BCAAT is present in other tissues, not the liver, but heart and muscle. And again, the liver, very small amount. Heart and kidneys mainly. It can be taken up by the liver and will undergo BCKAD and oxidation, potentially turned into fat, made into glycogen, like other amino acid carbon. Okay, so that's the fed state. Any questions about that? Okay, so noting glutamine, alanine, very important amino acids coming from the muscle in the fed state, and the keto acids and what happens to those in that normal physiologic, fed state. And the intestine is a major consumer of that glutamine. What happens in the fasted state? And I'm going to say this is the early fast, right? Because over time we'll adapt and we'll have less skeletal muscle proteolysis, as you adapt to a true starvation state. So this is kind of when the glycogen's gone in that early post-fast stage, what happens. So what happens is we'll get muscle breakdown, yielding amino acids, and we're talking about branch chain predominantly here. So valine and isoleucine go through the BCAAT activity, making glutamate, glutamine synthetase, all this is the same, alanine, right? All of this is actually the same. So it's what happens importantly that's different from fed to fasted is right here. And that you've got BCKAD activity now is 5-fold higher under these conditions, right? As I told you a moment ago, right? So now we have, before in the fed state, we had that opportunity for other tissues to take those BCKADs, the keto acids, and either reaminate them or use them in other ways, right? It allowed a fate other than immediate degradation, having that enzyme activity low in the fed state. Now, in the fasted state, and I said also in metabolic stress, okay, we're going to have a big increase in BCKAD activity, which now allows those BCKADs to be oxidized to the TCA intermediates that they yield. Okay, so in the case of valine and isoleucine, this acetyl-CoA and propionyl-CoA and succinyl-CoA, those are going to go into the TCA cycle and going to generate some energy for the muscle. They might be used for some other things, but that's the predominant thing. All right, and then what happens to leucine is very unique. Remember what we said about leucine is that it's uniquely ketogenic. It yields 2 carbon intermediates, right? Acetyl-CoA and acetoacetate. Those are highly conducive to being immediately and completely oxidized for energy. So we get a lot of energy in this condition from leucine. And it's, giving us ammonia that can be then combined with glutamate to make glutamine. Okay, so that's in the early fasted state. What's happening, and I'm just looking to make sure I haven't missed any major points here. Yeah, are there any questions? Yeah.
Can you go over again how BCKADs are regulated in the fast? So why do they?
Right, so BCKAD is activated by a kinase, right, that phosphorylates it. And that only happens when insulin is low. So in a fast, you don't have insulin. It's a low insulin to glucagon climate. In a metabolic stress response, you've got essentially insulin resistance going on. And that's why, okay, and there are things we can do to attenuate metabolic stress, and we're not going to talk about that really now. But for the most part, that's functional. It liberates protein and amino acids for you to do what you need to mount an immune response when you're in a metabolic stress response, right? These are highly catabolic conditions. So if you think about critical illness, it's a highly catabolic state. And that low insulin action at the muscle is allowing that to happen. It's functional, even though it doesn't seem functional sometimes, but yeah.
So does that happen in other types of insulin resistance too?
Interesting that you would say that. There are other things driving it. So it's not as simple as I'm putting it. I'm just trying to really focus, which is always the challenge, right? Because I want to give you a big picture. Alicia just asked a great question. Is it the same thing that's happening in other types of insulin resistance where you've got obesity-related insulin resistance, for example, or whatever? There are other factors in metabolic stress. It's not just the low insulin to glucagon. You've also got cortisol and catecholamines and cytokines, right, that are driving proteolysis. So it's not just that. But in terms of the enzyme activity, it is. So thanks for asking that, but it is a little more confusing. Yeah, Andrew.
This might be picky, but because there's not an arrow pointing to pyruvate, is pyruvate a product or being incorporated into the...
No, let me make sure. So I know this is hard when I write reactions this way because I've got the enzyme in the middle here. This is showing glutamate and glutamine. I mean, no, sorry, not glutamine. Glutamate and pyruvate are being transaminated to yield alpha-ketoglutarate and alanine. Okay? Is everybody following my? It's hard to draw stuff in PowerPoint. I don't know if you've ever tried to do it. It's really. And the same thing up here, transamination. These amino acids, are undergoing the transamination reaction with alpha ketoglutarate to yield glutamate and the keto acids, right? So they're kind of crossing. Any other questions about this particular? Okay. Here are the cofactors. Riboflavin, niacin, thiamine, pantothenate, coenzyme A. It's kind of cool. It's a very interesting biochemistry, chemically. If you take a grad level biochem class or a grad level vitamin and mineral class with Dan, if those of you that end up going on, you get to hear the whole story. It's very interesting, but I won't belabor it. Okay, that's it for muscle. We're going to move on to kidneys. Any questions about muscle before we do? So the study guides are posted, and God, for the life of me, I can't remember whether Dr. Chen asked about the cofactors. So can you just refer to the study guide? If we ask about the cofactors, it's not that hard to remember. I mean, riboflavin, thiamine, niacin, I mean, that's useful to know anyway. And if you think about it, there are a lot of vitamins involved in amino acid metabolism, right? B6, you can almost take a wild stab at it and you get it, B vitamins, they're pretty much all involved. But I'm not 100% certain if we asked about that. And I can't remember where Jessica is. Anyway, whoever knows the... Okay, let's talk about the kidneys. Actually, there are two of them usually. Now, there are more details on these slides than I'm going to call out, and you'll see in the study guide that I focus on some of the more important things. But the kidneys will kind of preferentially extract certain amino acids, or more amino acids than others. They take up aspartate, they take up glutamine, alanine, and citrulline, right? And citrulline is not an amino acid, but it's a derivative, and we've just talked about that, right? So this hopefully will be fresh in your mind. And the kidneys are a major site for the synthesis of histidine, arginine, and guanidoacetate. Let me talk about guanidoacetate here just because it's easy, and histidine I'll show you on the next diagram. I think I've got it. Actually, yeah, I do. So guanidoacetate, just again, a bit of trivia, but it's essential for creatine synthesis. Where do you think about creatine showing up? Creatine. Someone say it out loud, because all I hear is pss, 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 pss. Muscles. Yeah, great. Muscles, right? As creatine phosphate, that high energy storage that allows you to leap out of the way when the mountain lion is about to pounce on you or something like that, right? Creatine phosphate, high energy. So that's where guanidoacetate, just a little bit of, it's a trivial pursuit question. Creatine synthesis happens in the kidneys. Okay, we're going to talk a lot about, arginine, citrulline, these other things in a second here. Yeah, and this is animated. I'm going to click through it just because there's other amino acid metabolism. Lots of stuff happens in the kidney, okay? I'm not going to ask you about serine and glycine, okay? I'm trying to get down here to, I'm showing you citrulline and aspartate coming in. Those get made into arginine, and that's important. Arginine is used for guanidoacetate, but arginine is also, going to be exported for protein synthesis. That piece of it is the main piece that you want to keep straight. Arginine synthesis, so important for whole body protein synthesis. Here it is. Histidine comes from a peptide called carnosine. Okay, and so that's, if people ask you, why is it that there's this controversy about histidine and it's being essential or not, right? Well, infants can't really, they don't make carnosine to any great extent. Adults, we kind of can get by with cleaving carnosine and releasing histidine to some extent for some period. So that's where the controversy comes from, because there is some endogenous synthesis of histidine. You have to say it. Carnosine gets cleaved to yield histidine. Okay, so that's pretty much all I wanted you to know about that, just because we talk about when kidney failure is, their histidine is conditionally essential, and that's why. Okay, this is also highly showing glutamine and alanine coming in, what happens, and we're going to talk a lot about this in a more easy way. Okay, so this is just showing it here. And yeah, this is more convoluted than I'm going to click through this. You can just kind of skip this. You can write, basically skip. We're going to talk about the individual reactions, but it's very hard to follow on this slide here. So let's go to more easy diagrams to follow. You've seen this. I'm not going to go back over this unless you have questions, but this is, remember what's happening. Citrulline coming from the intestine, being used to make arginine, and how that happens where arginine is majorly released for whole body protein synthesis. And I've got it animated again. Any questions about this? Okay. The only thing I want you to know about this just shows you, what's going out. Nitrogenous compounds in the kidney, just FYI really. But I will say this, I want you to know this, that under normal physiologic conditions when you're eating at regular intervals, about 80% of the nitrogen that's going out in the urine is in the form of urea. In a fast, metabolic fast, extended fast, we start to excrete more in the form of ammonia, and that's where glutamine comes in. Okay? Glutamine is the transport that comes into the kidney. It gets cleaved by glutaminase to yield ammonia under conditions where you're in an extended fast. Okay? So under conditions where you're, in that fasting state, nitrogen excretion is going to turn into more like a 50-50 between urea and ammonia. And it's not exactly 50. There are these other things, very small amounts of this other nitrogen. But for our purposes, you can just think of it as increasing it proportionally. So more of it's going out as ammonia than under normal circumstances. And there's an important thing about that we'll talk about when we get to interorgan glutamine metabolism. Actually, on the next slide, I'll hit it as well. Okay, so in the kidneys, they become very important in a prolonged fast. The two reasons why are right down here. And I don't know how many of you knew that the kidneys have the machinery and capacity to make glucose, gluconeogenesis. You knew that. Great, I love it. Okay? This becomes a very important thing in this kind of extended fasts. The kidney actually is kind of 50-50 with the liver, actually, for gluconeogenesis under these conditions. Okay, so number one, number two. The number two here, okay, let me explain what's happening. Kidneys take up glutamine. They have the enzymes. I don't know why this isn't animated. What three key enzymes? We've got glutaminase, right? Again, anytime you've got glutamine being cleaved to yield ammonia, you need glutaminase. Sometimes we have a transaminase reaction, but here we're going to talk about glutamate dehydrogenase. So the kidneys are very effective at getting that ammonia streamlined and released from that glutamine to yield ammonia, which then acts as a proton sink, an acid sink. It basically takes hydrogen ions avidly, because that's what ammonia does. It forms ammonium, and in the tubule, when that happens, the ammonium ion will be excreted, and it's a way for the kidneys to get rid of acid. Okay? So glutamine metabolism in the kidneys in an extended fast is particularly important as a way of ridding the body of some of those acid, the hydrogen ions, right? So ammonia combining with hydrogen ion to ammonium, and it gets excreted. Helps us get rid of acid under these conditions. And helps to normalize the pH. So I've got two 50-50s, right? Under these conditions, remember I said nitrogen excretion kind of gets to be 50-50 with urea and ammonium. And then I also said under these conditions, the liver and the kidneys are working to produce glucose, kind of in a 50-50 ratio. Okay, those are the two enzymes, and then we have ALT and other transaminases, ALT being very important to yield pyruvate to glucose, but there are some others that are working as well. But thinking about alanine, transaminase producing pyruvate, feeding into gluconeogenesis, that's kind of a really important thing to keep straight. Any questions? Now, because I'm about to talk about this, as we move into interorgan glutamine metabolism, I want you to write one more thing up here. It's important in a prolonged fast. It's also important in a condition we call metabolic acidosis, where the blood pH is low, acidic. Metabolic acidosis. Now, it so happens that a prolonged fast is an acidic state. I'm going to talk about that more in a second. But metabolic acidosis can happen independently of a fast. It can happen in an ICU patient, for example, those people that are under metabolic stress potentially, a whole lot of clinical or medical reasons why somebody could be in an acidic state. Okay? So I'm going to just plant that idea here, and then I'm going to get back to it very soon after we move on here. All right. Is everybody with me? Hanging in there? All right. So now we're going to talk a lot about glutamine, and you're going to get tired of me saying glutamine. I'm going to get tired of saying glutamine. And hopefully I will not confuse you as I say glutamine in various ways. We've seen this, right? Remembering that the liver has an important division of labor around glutamine metabolism, different locations, different subcellular as well. But that avoids that potential futile, energy wasting cycling, right? So the liver really has an important role to play in glutamine metabolism, but there are other tissues as well, okay? And we're going to go back over kind of giving you some additional information, because some of it we've already talked about. So I'm just going to kind of wrap up our discussion about amino acid metabolism, showing the way that various organs and interorgan metabolism works. So glutamine synthetase, right? Paravenous hepatocytes, I said, are very, they've got a high affinity for any ammonia that might be circulating by. The blood leaves the liver through the central vein, so paravenous hepatocytes are kind of the last barrier, if you will, before blood leaves the liver and goes into circulation. So we know that ammonia is toxic, right? So it's really critical that the liver has those last guard sentries, whatever you want to think of, that are around that central vein, and they're taking up all the ammonia that might have escaped the urea cycle, or for whatever reason there might be actually something driving synthesis of glutamine under these conditions, right? So the liver paravenous hepatocytes have glutamine synthetic machinery. In large part, they're meeting the glutamine needs of the intestine and potentially supplying it to the kidney as well, right, in an extended fast or acidosis. And it's serving that function of the last ditch effort to protect the body from ammonia. If there's any ammonia around, it basically will make it into glutamine. Muscle has glutamine synthetic machinery, the glutamine synthetase, because it's basically dealing with ammonia from amino acid metabolism. And it is a major supplier of glutamine to the GI tract. And then the brain has it. And the brain, it's not a major exporter of glutamine. but it makes it from any amino acid metabolism going on in the brain, because as you might imagine, ammonia is highly toxic. We've talked about that. So the brain certainly doesn't want to have any ammonia around. And so if you think about the function, the fact that glutamine synthetase has such a high affinity for ammonia, that's a just really important feature of the enzyme. It's not going to let any ammonia hang around for any period of time. So there's no toxicity that's going to happen as ammonia gets released from amino acid metabolism in the muscle, in the brain, or in the liver for that matter. And I know you're writing, so I'm going to go ahead and click through here. On the other hand, we got glutaminase, and so the liver does have this enzyme as well, so it's got both, but it's got different cells that have it. And so glutaminase, its primary job is to release ammonia, right, for different reasons in different tissues. In the liver, it's to release ammonia into the urea cycle. In the kidneys, it's to release ammonia so that it can be excreted as ammonium ion, basically sinking acid, getting rid of acid. So it's under certain conditions it becomes real active. In the small intestine, it's always there, always active. The intestine needs it to use it for energy. And the same thing with immune cells, lymphocytes and macrophages. Okay, when you're done writing, put your head up for me. Okay, everybody got it? Raise your hand or shout when I turn the almost. Got it? Okay, no shouts. All right, this is the enzyme, high affinity for ammonia that makes glutamine, right? These are a review of points that, and some things I haven't said before. Glutamine readily diffuses in and out of cells, so that's useful. Readily taken up by any tissues that need it. Intestine, kidneys, immune cells. It's a major carrier of nitrogen between cells. We already told that. Most abundant amino acid in circulation. Primary energy source, I know you know this, for what tissues? Intestine and immune cells that we just showed you on the previous slide. It is an important, plays an important role in acid-base regulation. And we just talked about what's happening at the kidneys under those circumstances, so I won't repeat myself. I'll try not to, anyway. And then glutamine synthesis in muscle, important. Major supplier of glutamine to the intestine, right? Skeletal muscle being such a large, and it's mainly coming from branched chain amino acid in the fed state, and in the fasted state it's from skeletal muscle proteolysis, right? Certainly early in a fast, that's happening to a large extent. And then these are, I've got two slides, just kind of showing the major tissues, what they're doing. It's not necessarily showing you the nuance of different physiologic conditions, but I don't want to belabor this too much, but you can kind of see, if the paravenous hepatocytes are working on making glutamine, then that's going to, they'll ship that glutamine out so it can be taken up by, here it is, the intestine. Kidneys will be taking it up under certain conditions, what's happening in the muscle. And the brain's got glutamine synthetase as well to help detoxify that ammonia. On the other hand, glutamine utilization means glutaminase activity, right? And I'll just remind you of that reaction. And then here are the major tissues where glutaminase is working. And I'm not showing, I think it's animated down here, but in the intestine, just remember that glutamine metabolism is producing ATP for that intestine. So it needs all those enzymes that we talked about in order to do that. And this is going to be different physiologic conditions potentially at the kidney where you're either making glucose or other things that are going on. So this is not nuanced by physiologic state, just showing you organs. Now, and this is very much a review, so mostly the first few slides here. When we talk about the intestine glutaminase, right, important for energy, in so doing it gives it has a huge trophic effect. And uptake of glutamine from the blood So remember what I said, you know, you've got diet, and you've got blood feeding the intestine. Well, in an extended fast, it makes sense. You're not taking anything in by diet, right? So a lot more is going to come from blood, and it's going to come from the liver making, you know, providing it, and the muscle providing glutamine. Intestine has to have it. In metabolic stress and injury, it's a kind of slightly different scenario here. The intestine takes more. You may be getting fed too, but it's still going to take more from the blood, and that's because its need goes way up. The intestine is an immune organ. Okay, it's one of our big immune organs. It's got a lot of lymphoid tissue in it, and when the immune system is activated, like in metabolic stress, your glutamine requirement goes up a lot. Okay, so the intestine will suck up more glutamine from the blood under those conditions of metabolic stress and injury. All right. Still staying on glutamine utilization with glutaminase enzyme in the liver, right? Let's go back over. Remember that in the fed state, glutaminase is active, so are the urea cycle enzymes, right? So hepatic glutaminase is going to be breaking down glutamine and using that ammonia to make urea. That is the normal state of affairs, okay? Now, incidentally, and it is incidentally, it is coincidentally, I can say the same thing is happening in the condition we call metabolic alkalosis, okay? So metabolic alkalosis, it is a separate physiologic state from the fed state, okay, obviously, but the same enzymatic reactions are going on, in this case, talking about periportal hepatocytes, glutaminase, that action is increased, you know, it's active in metabolic alkalosis. That means high blood pH. And again, these things happen all the time in hospital settings. For the most part, most of us are not walking around, you know, with acid-base disturbances, unless you might be bulimic or anorexic or some things like that where that can happen, okay? Or you're taking medications that could cause it, okay? The significance, and actually I'm asking that question, the significance of this is that we need to be breaking down amino acid nitrogen that's coming in, right? So that glutaminase action and the urea cycle, if you go back to the liver section, you'll remind yourself how that's important in the fed state because we're trying to deal with amino acid nitrogen. So that's the significance of that. What's happening to kidney glutaminase activity under this condition? It's essentially shut down. So the kidney glutaminase is basically not active at all when the hepatic glutaminase is. So the liver and the kidney, we say this is sort of reciprocal regulation or interorgan cooperativity, if you will, that when the kidney, and I'm going to talk about the opposite state of affairs, when the glutaminase is active at the kidney, it's not at the liver. So that also is kind of a, you know, you don't want to be making a lot of glutamine. Well, actually, I'm not going to say that. I'll leave it simply. So under these conditions, kidney glutaminase not active, okay? Reciprocal regulation between these two tissues. And it'll become more clear now when I talk about kidney glutaminase. And in a fast and metabolic acidosis, so the exact opposite conditions as the previous slide, kidney glutaminase is going to be very active under these conditions. And the whole point is for it to get ammonia so that we can excrete acid. So under these conditions, again, the hepatic glutaminase is essentially not active at all. The urea cycle is not active at all, basically. And you've got these enzymes. I've just got to get them all animated here. So you've got glutaminase, getting rid of the acid here, trying to bring the pH back up. You've got glutamate. You've got ALT. Particularly important in a fast, you've got gluconeogenesis becoming really important under these conditions. and you need those enzymes to basically make that happen. Now what about glutamine synthetase? So we've just said the kidneys are breaking down glutamine. Well, it's really important that under those same conditions, the liver is cranking it out. The liver is working, this is called the intra-organ cooperativity between liver and kidney. In an extended fast, the liver is making glutamine so that it can be taken up by the kidneys, and the kidney glutaminase can act on it and excrete acid at the kidney and help bring the pH back up. So the kidney and the liver are working together with the opposite enzymes, right, active at the same time. Paravenous glutamine synthetase is active when kidney glutaminase is active in that extended fast, and also metabolic stress, but we'll focus on starvation at this moment. Similarly, acidosis, same kinds of thing, the same action is happening. And I just said the significance of it is this working, the liver are liver paravenous hepatocytes. Sorry, I got way too fast there. The paravenous hepatocytes and the kidney glutaminase, kidney cells, with glutaminase, they're working together. Now, acidosis happens in a fast because you've got ketone bodies being formed, you've got protein catabolism that is amino acid metabolism leaving, increasing release of amino acids, so you get that acidosis. All right, and I think I've answered this, so I'm going to kind of leave it and hope you can think about this as you come back to these. And I've got this depicted in a diagram here, showing the enzymes where they're active. Glutamine synthetase is active, kidney glutaminase is active in an extended fast in metabolic acidosis. Okay, so I've given it to you graphically as well as written out. And then this is kind of a summary for you. And so if you want to, and if you have to be careful if you do this, so you're going to write down extended fast and metabolic acidosis. They do not have to coincide. I said a fast is associated with acidosis, but you can have a full-blown and much worse acidosis independent of any fed or fasted state. So you can write it up here, extended fast. And metabolic alkalosis or fed. And it's not, again, please note, coincidental, but not necessarily coming together. And so how do we excrete ammonia? So how do we excrete nitrogen at the kidney? In acidosis and in extended fast, please write 50-50 between ammonia and urea, 50-50. It's more, more ammonia than normal, but it's still just 50-50. We're still making some urea. Alkalosis, it's like 80% of it is urea. And I think that the rest of it hopefully is self-explanatory as a summary kind of thing. And then the last final thought I'll have for you, those that are going on to medicine, those of you that are in MNT with me right now, the clinical applications of this information are important. Glutamine has had an enormous amount of money thrown at it for research studies, particularly in critical illness. And we have evidence-based guidelines for some conditions that would say, particularly trauma and burn injury, that it can be very helpful to provide additional glutamine. So that's the bottom line. But in metabolic stress, the intestine takes up more glutamine, the hepatocytes, the paravenous ones, they're working at it, but they can't quite meet the demand. The intestinal demand is so high. So you get this kind of relative deficiency of glutamine in circulation, and muscle catabolism is being driven, and it alters the function of the GI tract and immune cells actually to have that deficiency. So if we can give it, if we feed people, that does an enormous good, but if we actually give them additional glutamine, it actually does more good. Okay? So trauma and burn injury, there are critical care guidelines that say, yep, burn injury, it can be helpful to give glutamine enterally or in IV if we can. Bone marrow transplant, short bowel and inflammatory bowel disease, probably less firm guidelines on that, but they may be beneficial. And they basically, if you think about short bowel bone marrow transplant, that's an immune, we suppress the immune system, and then we're trying to help those cells take root and grow, right? Glutamine is a preferential energy source for those cells. Short bowel syndrome and inflammatory bowel, those are gut cells that are being affected. So if you give them glutamine, the thought is that they help to help the bowel adapt after we take out large pieces of it and other things like that. And I know I have talked to the absolute nth minute, right? I cannot believe I finished. If you have questions, I will stay. If you want to ask them now, please feel free to, and I apologize that it came right up to the nth minute. Thanks so much for having me. I appreciated having the chance to talk with you and, plug some of the relevance of biochem to clinical nutrition and other parts of nutrition. So, your study guide is posted, so please use that. Those of you that know me know that I won't do anything that's not on that study guide. And if you have questions, just please ask them. Yeah. Hey, those of you that are leaving, if you don't mind just kind of keeping it down a little, I'd appreciate it so I can answer the questions. And those of you that want to stay, please feel free to. Yes. I worked with Dr. Chen to write the exam. Her question, okay, the question is, did I write the exam for this section, and how much of this exam is this section, I believe? And please, is there a TA here? Yeah. It's all this, right? Yeah, it is all my material. So the next exam is all my material. There is a quiz that's write-in, right, coming next that is based on the study guide. I'm pretty sure she has a quiz and then she has an exam. And much, right? Are you all shocked by that? No? Did she, sorry? Next period is listed as a review. Okay, well, yeah, no, I'm not saying next. I'm just saying at some point, I think you might have, no? Maybe she's not going to do a quiz? Oh, okay, well, great. You don't have any write-in then, I guess. I don't know. Last year she did a write-in. Okay, so the exam is multiple choice, matching. Use the study guide. Did I answer your question? Okay. Good luck.